And thanks everybody. Welcome to this way of meeting each other for the first ID-PRISM meeting with our students and with our faculty. And first of all, and I'd like to introduce a bit what the ID-PRISM Center is, and give a very brief introduction on what we want to do this, and what's the purpose, and what's the organization, and what's the content, and what we would like to do. So I assume everybody can see my shared screen. And so the ID-PRISM stands for Infectious Disease Prevention Research via Innovative Statistical Methods. And there is a link to the website, and now it's a little dimmed, but you can see that. You can click that link to, it will lead you to the website. Next. So our mission for the ID-PRISM Center is a collaborative research and educational effort to foster collaborations to rigorously answer scientific questions in infectious disease research via innovative statistical methods modeling or mathematical modeling or other quantitative approaches with an emphasis on prevention interventions. We're not saying we're not interested in therapeutics, but we have a particular focus on prevention research. And we would like to provide more training and educational opportunities to our trainees or to our students or RAs in independent studies or in doctoral dissertation or master's thesis or capstone project supervision, and also in terms of providing research assistantship and undergraduate student internships. So our members for the ID-PRISM Center mainly include, but certainly not limited to, our Hutch and UW Biostats, Statistics and Bioinformatics faculty, and or statistical research associates and assistants and students, both graduate students and undergraduate students. So the main activities we are thinking about for the ID-PRISM is monthly meetings rotating between the Hutch and the UW. So the meeting agendas, we are expecting them to be diverse, to promote center objectives, and will be in the form of seminars, state-of-the-art quantitative methods research, and or applied data analysis, spanning from theoretical-oriented work to applied work. So it doesn't have to be completely finished manuscript or completely finished work. It can be work in progress, talks or discussions, for example, related to your doctoral and master thesis, draft manuscripts, or RA projects. And we would like to provide more training opportunities by linking between students and the faculty in terms of, for example, implementing computer code or package, or sometimes with data processing, data management, data cleaning, and data collections, etc. So the upcoming activities, because apparently the very serious situation related to COVID. So there are a lot of challenges presented by this novel coronavirus and the COVID-19 diseases. So we are thinking about the topics focusing on the broad spectrum of the research questions we are interested in terms of answering those questions related to the novel coronavirus. In terms of biology, for example, virology, how the pathogens is affecting humans, and how come they're only affecting a portion of people, but still with very diverse disease symptoms. And for example, we are interested in epidemiology, how the outbreaks are happening around the globe. And we are interested in disease natural histories, and we're certainly interested in diagnostics testings and also surveillance testings. And for example, we're also interested in treatment. What kind of therapeutics are the candidate regimen for treating infected patients in terms of how do we rigorously answer the questions via statistical study designs, data collections, management, study monitoring, or even meta-analysis and analyzing multiple studies at the same time. And also we're interested in preventions, and we're interested in non-pharmaceutical prevention interventions like social distancing. And now today, CDC advising maskings. And also how about pharmaceutical prevention such as vaccine development? That's probably the holy grail for the prevention strategy. And also how about the PrEP? How about we give the drugs, if we prove they're therapeutically effective, can we use them for prevention? And how about the PrEP, post-exposure prophylaxis? So about prevention, and certainly we are interested in statistical modeling and mathematical modeling and bioinformatics analysis. So those are all the upcoming activities we're thinking in the next few months for our topics from ID-PRISM. And today this is our first inaugural meeting with our students and the faculty together, and we are presenting an overview of ongoing ID-PRISM COVID-19 activities. And Alex, probably can share agenda and talk about that.
Yeah, sure. Thanks, Ying. Yeah. So we're very happy to have such a large first meeting. So this is, I think, hundreds of students from the two departments, including welcome to the Biostat admitted students who are on the call as well. So I won't take up much time because we have a lot of speakers. I just wanted to take this opportunity to say to the students sort of how incredible of an opportunity this is to be in Seattle right now doing biostatistics and statistics research. So our list of speakers today is quite impressive. I mean, there are policymakers here really at a global level. So a couple of our speakers, our first two speakers, just published a paper in the New England Journal of Medicine suggesting, and they'll speak much more about it, but suggesting an approach to sort of manage all clinical trials for COVID-19. And there are sort of many other researchers from the UW and the Hutch who are excited to present to you and also excited to potentially have you involved in their research. So please don't hesitate to reach out. Speakers where possible will point out areas for potential student involvement. I want to take this opportunity to thank Ying, who's the current head of ID-PRISM, Peter Gilbert, who's put in a huge amount of work to put this all together, Marco Carone, who's helped quite a bit to coordinate with Biostat, Adam Elder, a student from Biostat, who's done a ton of work to get this meeting together, Kendrick, who will be moderating most of the discussion, and then Christine from statistics and Kira from the Hutch for putting together such a large meeting. It's really been a lot of work. This is just a brief reminder. Speakers, please stick to your time. I know 3 minutes or 5 minutes goes by very quickly, but there are so many. I just want to make sure that everyone has a chance to speak. And as a final note for all participants today, we will be posting meeting minutes on the ID-PRISM website within a day or two of this. So there are students actively taking minutes of the whole meeting, and the meeting's also recorded. And so if you miss some things as the meeting's going on, just please be aware that there will be minutes posted. And so with that, I'll pass off to Marco to give a brief intro from Biostat, and then we'll get to the speakers.
Marco, you're muted.
Marco, mute. Sorry about that. Thank you, Suzanne. So no, I was going to say that. Well, thank you, Alex, and thank you, Ying. And I'm just going to say just a few words because I think that the introduction is great and description of what the center is about is comprehensive. We've had for many years now a lot of interactions between UW and the Hutch in this broad area of infectious diseases. And of course, the center is new, and it provides, I think, an infrastructure to be able to organize events and have a clear presence that the students can see and join in. So in that sense, it's really meant to focus our energy and be able to be out there and attract many students. And I'm glad that the students have been coming in great numbers. You can see from previous years, other students that have gone through this path, even though we didn't have a center, and we're at UW in stats and biostats both, doing dissertation work with faculty members, again, in stats, biostats, and at the Hutch in this broad area. And what I'll reemphasize, something that you mentioned, the work really does run the entire gamut from theoretical work all the way to applied work. And that's by design, and it actually serves a purpose. And so regardless of where at any given time your interest might be with respect to the statistical sort of theory to practice spectrum, what ties us together here is an interest in making a difference in infectious disease research and practice. Of course, now I think it's much easier to convince people that it's important. It's very timely, but you can contribute in so many ways to this area. And so, again, we have a track record of excellent students having gone through this, having been extremely gratified by the work they've done, and having had a lot of success moving forward and continuing to make contributions afterwards. So anyways, I'm very proud of that. I'm very excited to see this coming together. You know, we're all in biostats certainly very vested in making sure that this center continues to be a focus for the many years to come. So that's all for me. Of course, I'll be happy later on if there are questions from the biostats perspective to answer them.
Okay, Kendrick, and so with that, we'll pass it off to you to begin moderating. So speakers, I think Kendrick will say this, but he'll let you know when your time's up. And I would just ask, because we have so many speakers, to please pass the mic when that.
Okay. Yeah, thank you, Ying, Marco, and Alex for the great introduction. So my name is Kendrick Lee. I'm a fourth year PhD student in UW Biostats. I'll serve as the student moderator of this meeting. But honestly, I'm just the annoying person who will tell the speaker the time is about to run up. So to avoid the awkward situation, please be mindful just to remember the time. And also, I think three of you have emphasized enough how important it is. For me personally, this is also a great opportunity to learn about how UW Biostats and Fred Hutch is involved in this global situation. So it's a great opportunity to learn of those research topics. So just a few other things to emphasize. I think we will have a 10 minutes Q&A session at the very end. But I guess if anyone has any questions, please feel free to unmute and then speak out. And I guess that's it. I guess I'll let Elizabeth Halloran take over with her talk about population-based assessment of epidemic spread, intervention, mobility effects, policy advisory panels, social distancing.
Or what? Good, thank you.
Yeah, no problem.
I really appreciate this off. Given 10 minutes, I'll be a little short than that. A couple of the other speakers are going to be talking about things that I've also been involved with, so I don't need to spend much time on that. Just to introduce myself, in case you don't know me, I am a regular professor at the University of Washington in the Department of Biostatistics. I am also a full member at Fred Hutch in biostatistics, bioinformatics, and epidemiology. I'm also now currently the interim head of the program, and we call it BBE, at Fred Hutch since January. I'm also the director of the SISMID, which is the Summer Institute for Statistics and Modeling in Infectious Diseases. And I founded that in 2009. And I thought, and that it offers a lot of really interesting courses related to the things we're talking about. And the good news is that I got a really good score on the grant. I found it out yesterday. And so it's likely that it will not die, but that it will continue for another 5 years. So you'll be able to take courses in that. This summer, of course, we'll have to have a reduced we cannot have people, 7 of my instructors currently cannot enter the country, but we will be doing something. So that SISMID is a great opportunity to learn more in this field. And then I'm also the director of the Center for Inference and Dynamics of Infectious Diseases, which was funded by the National Institutes of Health from 2014 until now, but they changed their mind about wanting to have centers of the kind of important work we do. And so I'm not sure what's going to happen. So to proceed, so a lot of the work I do right now is with a colleague of mine, Alessandro Vespignani, who's at Northeastern University. And they do these big global modeling of infectious diseases. They have a platform that they built that is able to, they have all the air traffic data from the entire world. And so we have worked with them since 2014. He's also an instructor in the Summer Institute since 2010. And we've worked with them to model Ebola and Zika and now the coronavirus. And so it's a big model that can also be fit to data. So we've had a paper in Science looking at the spread from China. We've had, and how what the travel restrictions did. We had a paper that just came out yesterday in Lancet ID that was estimating some of the important natural parameters, natural history parameters, based on also data from China. And in case you're interested, a lot of the data from China are available on websites. It's publicly available, and you just have to know exactly where to find it. And we are also, and this is now not totally formally announced yet, but he's one of the modeling groups that was chosen by the White House to do the modeling for the White House. And so you've heard a lot about the IHME model, which is a very simple model. It's a Gaussian things fit to some data, but we do actually transmission dynamic modeling and can look at interventions. And so right 5 minutes before this call, he sent me the prototype website that they're gonna open up for the White House modeling. So that'll be something to watch. So I want to talk, so that's where a lot of my work is now with the team there, because I don't have a big team anymore. I'm sort of not, at a different stage in my career. But I want to talk briefly about the WHO work, the World Health Organization, WHO, and a couple of the people coming after me are going to talk about that. But after the Ebola outbreak in West Africa, where nobody in the world was to do or think about, I mean, it was just everybody was totally taken off guard. There was a movement called the Blueprint for Research and Development, which developed different work streams. And one of those work streams, study design. And so this is something that maybe Tom will talk about, the New England Journal paper that just came out. That was a 4-year project in the works, at least 4 years. But that's part of, and then Yunda and Holly, somebody's going to be speaking about the core protocol that we've been working on, or they've been doing more work than me, but I'm on these WHO committees and part of that. So that's an important area where we make international contributions. There's 2 final things. I've been asked a lot to think about the end game. I mean, so we're in lockdown now. And so what happened? Are we gonna stay like this for 18 months while people hope there might be a vaccine? And so the question is, what's the end game? And I've thought a lot about that and talked a lot to reporters. The end game is where we could do modeling or statistical estimation. Things like, well, if we have broad scale testing, cheap and efficient, you can test yourself to see if you're currently infected, even if you're asymptomatic. So we need to be able to do the serologic tests of everybody. So how many people on this call out of the 856 have actually had coronavirus? And we need to be able to know that. Personally, was sick. I got sick 2 weeks ago and I got a test and it was negative, but I'm pretty sure it was a false negative. And so I want a serological test because I have not left my house in 2 weeks. But that's okay. So, and so then another thing that we're getting involved. So those are kind of questions. So how can we do this and not just all stay in our houses, but if we can test people, isolate people quickly, isolate their contacts. And so some people are interested in learning how to work with mobile data. And there's a lot of mobile data available. And people are now thinking about how are we going, and I'm only going to say a couple more sentences and I'm done. How can we do contact tracing without actually sending to people's doors and doing that shoe epidemiology? And so there are people now, and you might look at Next Trace, Trevor Betting on this, people at Oxford, people in other places. So how can we do a registration system where, well, I just tested positive, I'm going to go online and I'm going to let you look at my mobile app, my mobile phone, and see where I am, and maybe dig up the people I've been contacting in the last couple days when I didn't have symptoms. So, or, you know, I tested positive without symptoms. So a lot of opportunity to work with a lot of data in a lot of different ways, contribute to dealing with this break and other outbreaks. I think that's what I have to say. That was funny. I don't know if there's any questions right now, but otherwise we'll go on to speaker. This Tom has this fancy background. Looking at my kitchen, or part of my kitchen.
Okay, so is there any questions from the audience? Okay, thank you for the great talk and the great overview of what the WHO grant entails. I guess our next speaker will be Tom. His talk will be addressing the COVID-19 pandemic with core protocols based on learnings from EBD.
Great, thanks Kendrick and Alex and Ying for putting this together, and Peter and Marco. Can you see the slides as I'm trying to project them here?
Mm-hmm.
Okay,
yeah.
Great. So I wanted to follow up. It's been a privilege working with Bets and the WHO R&D working group that Bets referred to. What I'd like to do is kind of expand a bit on what Bets is saying and set a little bit of the framework for some of what others are going to say after me by reviewing in one slide kind of how we got here from an R&D working group and set the stage for our response to COVID-19, and then a little bit about what that response is. So going back a bit, in 2014 and mainly in 2015, we had the major Ebola outbreak in Western Africa. And there were big debates about whether or not in clinical research can't do randomized clinical trials in public health emergencies. And many of us believed that it was critically important to do so. We needed reliable evidence in those settings. For Ebola, optimized standard of care like hydration itself can reduce mortality from 90% to 50%. And so there were a lot of discussions in the time period around in 2016, the National Academy of Medicine and WHO took these issues on. I had a chance to work with both groups, and I'm delighted they both strongly endorsed that we need randomized clinical trials in public health emergencies. And an excellent such trial was PREVAIL 2, which was randomizing to a monoclonal antibody, ZMapp, against optimized standard of care. And it had great characteristics. It was randomized. They were using an optimized standard of care, and it had a mortality endpoint. Fortunately for the public, not for science, there was a waning of the outbreak when the study only had 71 of the intended 200 patients. So there were lots and lots of interactions with Bets and myself and others saying, how can we be better prepared so that when we get public health emergency outbreaks, we're in a better position to be able to do evidence-based research. And we were widely interested. We were at the time, obviously, still very keenly interested in Ebola, but there was a lot of interest at the time in Zika in Brazil and in South America, in Lassa virus in Nigeria, chikungunya that was in India and Africa and in Asia, and Nipah in India, and of course, SARS from the outbreak that we'd had in Asia, and MERS, a more recent outbreak of coronavirus that had occurred in the Middle East. And I think we were better prepared because in August of 2018, we had the outbreak of Ebola that emerged in the Democratic Republic of Congo, in the Congo. And it basically was a 12-month experience. WHO was amazing. They developed the concept of having compassionate access while infrastructure was being developed, and then RCTs followed. And so as the outbreak expanded from Beni to Batumbo to Katwa to Mangina, we were able to get half of all of these infected people onto these scientific studies. It was an amazing accomplishment. And I was the chair of the International Committee for Murray, for the compassionate access. And we argued that anybody who was eligible for the RCT was no longer eligible for compassionate access. They had to be channeled into what was called the PALM-RCT. And it was amazingly successful in that we were able to randomize not 70, but 700, and got reliable evidence about two monoclonal antibodies, MAB114 and Regeneron, as being superior. And our monitoring committee at the end of that 12-month experience argued that what had been done was not just important for Ebola, but for the future of public health emergencies, that it created a paradigm for setting up infrastructure in a timely way to provide compassionate access and then channeling people into research to be able to get reliable evidence. Not everything was perfect, as Betts and I struggled through all of this. Note that the PALM-RCT had a ZMapp control arm. Well, the PREVAIL RCT only had a few extra deaths, 5 extra deaths on ZMapp versus optimized standard of care. But you release the results and the world responds. And there was such a response that we couldn't, in fact, use optimized standard of care, even though now that the data are in, it's not at all clear that ZMapp was much, if any, better than optimized standard of care. So we learned a lot, but we got weaker evidence than we would have if we'd had a proper control for the effective regimens. And we don't know what remdesivir does, which is unfortunate because remdesivir is one of our leading candidates right now in the COVID-19 setting. So as Betts mentioned, our article appeared yesterday in the New England Journal, basically saying we need a new concept, a new paradigm, which is a core protocol to be used in the future, including in SARS, in the current outbreak. So if I can scroll ahead here to the next slide. So in essence, for the last part of time I have, what are we doing? So basically, we have worked together under the auspices of the WHO and developed both a solidarity core therapeutics protocol and a vaccine protocol. Betts and I and others have been involved in that solidarity protocol that's now underway, looking at whether in hospitalized patients we can affect mortality. And the core vaccine protocol, I'm so delighted that Yunda is on here today with Peter and Holly and Betts, who've all played a very significant role in moving us to a point where we're closing in on being able to launch the core protocol for vaccines. And at the last half of the slide, many other things are happening too. There are developments of therapeutics broadly in pre-exposure prophylaxis and in post-exposure prophylaxis, pre-exposure prophylaxis in healthcare workers, post-exposure in household contacts. We'll hear some from Elizabeth, and who's been providing leadership along with Gates Foundation in setting up that PEP trial in household contacts. They're looking at hydroxychloroquine. Others are interested in chloroquine and the combination of lopinavir and ritonavir. And there are efforts now starting in early diagnosis. Also, the UW Gates Foundation key in that, looking at studies in early diagnosis and in hospitalized patients beyond what Solidarity is looking at. There's interest in IL-6 inhibitors, tocilizumab and Kevzara. So this is what's happening. What are some of the open questions? What are the struggles? That's kind of what I've shown in yellow here. So going up to the vaccine protocol, I was on for 4 hours this morning, typical day here with World Health Organization in Geneva and other colleagues internationally, trying to get at key design issues. What's the endpoint in a vaccine trial? I think we're saying it's virologically confirmed COVID-19 disease. Yet what we really want to prevent is severe disease and death. And can we presume that from showing that we can prevent infection? Big question, what do we have to rule out? Do we have to rule out no efficacy? Do we have to rule out moderate efficacy? And if you rule out simply that it's completely ineffective and you have only a very modest evidence of effect, disinhibition could readily offset those benefits. So big debates about that. In the Solidarity Therapeutics, with strong emphasis from and leadership from folks like Richard Pito, it is a mortality trial, yet we have lots of issues about how to optimally do the research. Remdesivir, as I mentioned, that was in fact involved in study in Ebola. Again, lopinavir and ritonavir, chloroquine are all agents, and there will be other agents. How do we efficiently add and delete agents from a large core trial? And then in these other therapeutics that we're looking at, what should be the endpoint in PEP and PrEP? So certainly we're leaning on infection, but severe disease and death, again, are what people really care about. And how do you know you've got the lowest effective dose? A major problem we're going to have is if you have an effect, you're going to want to be able to apply that intervention, whether it's a vaccine or a therapeutic, to millions of people, which is a supply issue, which means you better know the lowest effective dose just to help supply. And what is the endpoint in early diagnosis? It seems to be a hospitalization, but again, what we really care about is preventing people progressing to needing ventilators and having an effect on mortality. And the last example down in the yellow at the bottom is in hospitalized patients. For some of these IL-6 inhibitors that might be effective in countering the cytokine storm, it's a really an interesting phenomenon where patients that are at high risk, that ultimately at risk for mortality, seem to linger, improve, and then often crash. So can agents like tocilizumab and Kevzara, which are immunosuppressive, stop that crash? And yet at the same time, by being immunosuppressive, they put you at higher risk. So you can't skip phase 2. And if we have to go to phase 2, we need a quick answer. How do we efficiently do phase 2/phase 3 trials? So in yellow, we've got a very significant number of ongoing challenges methodologically. I can't help but pass on the chance to pitch. And by the way, Biostat 524 is just starting this week. So for students that haven't taken it, it's a great time to come and join us, where we're going to talk about all these methodologic issues that are really important for us to address. So it's one thing for us to be involved in the collaboration with colleagues locally and internationally to get studies in the field in a timely way for vaccines and therapeutics, where we can actually get answers when we need them. We don't want to be here in this social distancing paradigm for 18 months, which we may well be, and Bets will tell us because she knows the modeling. But we want to get timely but reliable insights. And so we need insights from everyone. I'm appreciative of all those who have joined in, and I'm delighted for many of others of you who will join in. Thank you.
All right. Thank you, Tom, for the great historical overview of the WHO core protocol. And thank you for advertising for Biostat 524. I took this class personally. It is a great lecture. So, okay.
Thank you, Kendra. Your $20 is coming shortly here. Thank you.
Yeah. So our next speakers are Barbara Richardson and James Hughes. Their topic is Seattle Coronavirus Assessment Network.
Okay, can I share my screen here?
Yes, please.
I'm told that I'm disabled from screen sharing.
Mm-hmm.
So that's fine. I just had one slide, but I can easily talk through it. So
I made you a co-host, so you should be able to share it now.
let me try it.
Mm-hmm.
There we go. Okay.
Okay.
And there and there. Yeah. Okay. So actually, I feel like I'm kind of a bit of an imposter here. I've been working with the Helen Chu and the Seattle flu study, which has kind of morphed into this scan study, but I haven't been really closely involved in the scan study per se. So I'll just tell you a bit about it. Really interesting surveillance study that's being done in the Seattle metropolitan area, where basically people can sign up online. There's like a daily quota. I checked this morning, and it opens up at 7 p.m. tonight. And apparently they typically fill up the number of people within like an hour or so. So they accept volunteer samples, people signing up online. And the sampling is, people are accepted into the sample based on where they live. And so this is through a census designation called public use micro data areas. And so you sign up online and a swab is sent to you for self-collection, nasal swab for self-collection, it's returned to the lab. It's been going for about 2 weeks now, and there's been over 2,000 kits sent out to people so far. The primary purpose of the study is to assess prevalence of disease, and particularly because it enrolls both healthy, it solicits people that are both healthy as well as people that may think they have COVID disease. It will get some idea of asymptomatics. and as well as severity and distribution of disease. And eventually, the hope is that these data can be used to assess the impacts of closure and distancing measures that have been going on. So be able to assess trends in over time as well as in geography, and by looking at only symptomatic people. The data is used, I'm told my internet connection is unstable, so I hope that I haven't lost you. Anyway, so it's being used to inform contact tracing by Public Health Seattle King County and their control efforts. Initially, and this is put together very quickly, it's primarily providing basic descriptive data, but the data will also be extremely useful in informing modeling efforts and predictions from models. And I think there's some interesting statistical issues about, you know, how to best evaluate and validate these complex nonlinear dynamic models of epidemics. And so I think there could be some interesting statistical work there. At this point, I know there are medical students involved in the study, but none from Biostat that I'm aware of. So I think I'll just stop there, and yeah, happy to entertain any questions.
Can I say something?
Yeah, go ahead.
Yeah, so I got sick Saturday 2 weeks ago, really frustrated. And I know somebody involved with the study, so I wrote to that person on Sunday. I couldn't get into the Seattle flu study. They said, oh, we're overwhelmed. But the scan opened that Monday morning, and actually I must have been one of the first people who registered for it.
Oh, good for you.
So I registered, I got in Monday morning. A couple of hours later, the test appeared at my door. I did the test, sent it back. The post person picked it up that afternoon, and the Tuesday, they said it had arrived. And then I didn't look for several days, but at the end of the week, I picked up my test, and it was negative, okay? So this is why, and I know some other person, a friend of mine had a friend who the same thing happened, it turned negative. So, you know, what was it? So now that's why the serology becomes interesting. But the test was negative. And if another, so I think it has something like, I heard 30% negative, false negatives. These are really important statistical issues involved here. What's your sensitivity and specificity of the test?
Right.
But then, you know, so how do you find out? It was my first, I was on the first day that they started scan. And
interesting. Do you know what the status of serology tests are at this point?
They're being developed. They are being developed. Yeah.
But none have been approved yet, right? Is that true?
I heard there was one approved. I read in the newspaper
yesterday.
Oh, yesterday. Okay.
Yeah, Bex and Dixon. Yeah, we're trying to look at getting it into Mombasa, trying to get into Kenya. But
people at Fred Hutch are developing, you know, this is for those of you biostat and stat students, developing a diagnostic test is not that complicated if you're a lab person. And so there are people at Fred Hutch trying to work on serological tests. I know immunologists all over the country who are trying to do that. And somebody told me the FDA had actually washed their hands of approving serological tests. And they're more interested in the other. Anyway, sorry to interrupt, but that's my personal
That's great. And also like to acknowledge that Barb Richardson is on the DSMB for several of these, the Seattle flu studies. So.
Okay, thank you for the short introduction of the scan study, Seattle scan study. And thanks to Elizabeth for sharing your personal insights from your personal experience. That is very interesting to hear. Our next speaker will be Laura Machad. Sorry if I pronounce your name wrong. The topic would be modeling of effectiveness of physical distancing interventions delaying the epidemic or flattening the curve.
Hi, thank you. I'm trying to see how I can share here. Okay, so I'm gonna talk to you about the other extreme of modeling. So Betts does this very complicated modeling, and I did It's very simple modeling. Basically, it was like started as a weekend where I was very frustrated with Seattle public schools not closing. And so I wanted to investigate originally what was the effect of that. And so what you're seeing here is, there are several interventions. I'm gonna describe them to you in a minute. But so basically, this is a very simple deterministic SEIR model that was calibrated with demographics of Seattle with 10 age groups. And so we, with the help of my postdoc, Tiffany Long, we investigated 4 possible interventions. So the first one is social distancing the high risk groups, and that's people over 60, and only them at night, and reducing their contacts by 95%. The second strategy we thought of was, okay, what if we do same thing with older adults, and then we also reduce the contacts in children. The third one, and that's the red curves that you're seeing. The orange curves is this a strategy that we thought of, like, well, what if we do 60 years and old, and the adults? And the rationale is always that you always want to protect the high risk people. And so the 60-year, like the older adults are always being protected. And then the purple one is this all. What if we, and that's kind of what we're doing now, social distancing of everybody. And so these panels, the top row is considering adults that are reducing their contact rates at 25%. And the middle row is at 75%, and the bottom row is at 95%. And so then we said, well, what if we investigate different timings at which these social interventions start? And so the one that you see, this column on the left, is if they are started 50 days after the first case was reported. And we chose 50 days because that's approximately what happened in Seattle. So if you consider the first case to be the first case reported in the United States, which was the first case was reported here, north of Seattle, then pretty much, I think it's 53 days that the governor enacted the first policy. And so what you can see, and this is 80 days, and the right row is if you start, the right column, sorry, is if you start at 80 days. And so what you can see is that this phenomenon of flattening the curve only happens when you start your intervention later on. But if you start early, as we did in Seattle, you're rather delaying the epidemic. And so this was all my time, so I'm gonna stop there. Thank you.
Thank you, Laura, for the short introduction. Is there any questions?
And so maybe I'll jump in here just for a moment. So I think because we have so many speakers, we may hold all remaining questions to the end.
Okay.
And then I think
I did want to know, like, didn't we skip Elizabeth?
Yeah, that was
Thank you.
So the other reason was we're going to go to Elizabeth Brown now.
Oh, I'm so sorry. Yeah, that's my bad. Yeah, I guess we'll let Elizabeth Brown take over. Her topic is designing COVID-19 prevention and treatment efficacy trials in the face of a rapidly evolving epidemic.
Thank you. There, can you all see my screen?
Yep.
Yeah. Okay, and then I'll be here too. Okay. I just wanted to share the picture of my skeptical dog because I feel like we all just need more animal pictures in our life right now. So there you go. She's looking at you like she doesn't believe what you're saying. So I've actually been incredibly busy in the last 3 weeks. It's kind of funny. I feel like the last 3 Sundays, I've gotten a phone call from somebody asking me if I can design a trial for them for COVID. So I'll talk a little bit about these 3 trials, mainly focusing on 2 of them, but I'll start with the work on the one that I'm not focusing on there. So I'll give a little background on this group called the Infectious Disease Clinical Research Consortium. So this is a grant that I applied for last January, and it's the leadership group is at Emory, and it's funded through NIAID, and my role is the director of the Statistical and Data Sciences Unit. So we just started meeting, or we started funding in December last year. We had our first meeting in January, and it's really funny to look back on that meeting. So Tony Fauci came and talked to our group because he was really excited about having this new group, but it was right when all the data was coming out about this, or the news about this new coronavirus, and there was a lot of talk, what can we do? And Tony couldn't stay long because he had to get on a plane to go to China or have calls with China to talk about what we could do about this. And it's really funny to look back and think about all these infectious disease experts sitting around the room together thinking, what can we do? Can we do studies on this? We're never going to be able to do studies in the US because we're never going to see it here. And here we are 3 months later, not even, and it's everywhere in the US, which is not what we were expecting. So that group is heavily engaged in COVID-19. It was supposed to be general infectious diseases and STIs, but instead it's pretty much our only focus is COVID-19 right now. So you've probably actually heard of some of the studies that this group is running, and that includes the phase 1 vaccine study that's at Kaiser, with some other phase 1 studies that should be starting soon. The big remdesivir trial that's been in the news is also an IDCRC trial, and we have a couple of sites locally that are running that, and that's a treatment trial. And we're currently, and this is what I'm most involved with, I'm working on designing a seamless phase 2/3 adaptive vaccine trial. And as I say on the bottom, I'm not really going to go into a lot of detail about this today because much of it's still preliminary, and we're covered a lot by NDAs with this because we work with pharmaceutical companies and intellectual property. Instead, I'll focus on the other 2 trials I'm involved with. And Tom mentioned one of these, which was the post-exposure prophylaxis study. And I'm also working on a treatment trial, and these are both with UW and funded by Gates. What's really different in these times is protocol development is fast. So I told you the last 3 Sundays I've been contacted, the first of these protocols is already enrolling. So you move through these very quickly, much more than usual protocol development takes months. And then on top of that, you're doing sample size calculations and study design without really strong evidence to support your hypotheses, your design, your power, your sample size, because we don't really have complete evidence yet to inform these trials. So, and for some things we don't have evidence at all. And sometimes we have contradicting evidence. It's because we're still in the early stages of scientific discovery. The other feature is that we're also repurposing existing medications. So Tom mentioned hydroxychloroquine, azithromycin, which you might all know is an antibiotic, also has some antiviral properties. They're looking at HIV treatment drugs. This means too, these drugs are already out there. Nobody's making placebo-controlled dummies for them. So it's very challenging to do placebo-controlled trials. And that means we have to do things like randomizing a whole household so that when you have another person who's either in your treatment trial or your post-exposure prophylaxis trial, they're not looking at each other's products and thinking maybe, well, one of us is on placebo, so we should share, or any issues like that. So that's kind of the best we can do for placebo control at this point. But that also means we have possible contamination of off-label product use because these products are, I would say, widely available. Hydroxychloroquine was, but people are hoarding it, so it's not as widely available. And a lot of producers aren't allowing production for outside their own countries anymore. But these both create a lot of issues or interesting design issues statistically. So as I said, not having a dummy leads to this household randomization for preserving blindness as much as possible, which induces correlation. Off-product use can lead to bias in our primary efficacy endpoint. So how do we handle that? Can we do post-hoc corrections on this? And the paucity of data to support trial design leads us to this adaptive design scenario where we're really redesigning studies as they're going on. And so we have to think carefully about what our plan is ahead of time for how we might plan to incorporate emerging data, both from the trial and from outside the trial, as we redesign these studies while they're in the field. And also, as I said, right now we're repurposing existing pharmaceuticals, and we might be looking at new ones that come up that are designed just for COVID. And so we have to be open to adding new interventions to these trials as they're ongoing, so that we're not restarting every time something new comes along, because it's very urgent to get high quality, well-designed trials done as quickly as possible, so that we can start getting interventions out, especially in the developing world, where I think this epidemic will hit much harder than it has for us. So I think I hit my time mark, but if anybody's interested in hearing more about this, I'm going to be talking to my research group about it in our bi-weekly meetings in 2 weeks. So you can email me, and there might be an opportunity to stop in on our Zoom call and have some coffee with us and hear more about it. Thank you.
All right. Thank you, Elizabeth. So maybe we'll, yeah, I think I may have been muted. so maybe we'll ask to Yifan Zhu and Ying Chen, who you've already heard from, on the analysis of early phase COVID-19 outbreak in China.
Okay, well, thanks, Alex. And so Yifan is not feeling well, so I'll step in to talk on behalf of him. And actually, this we did this project was because Elizabeth mentioned, 3 months ago. Now it really looks like 3 months is like a while ago. But when we actually, in the middle of January and toured right before city lockdown on January 23rd in China for Wuhan city, which is was the epicenter for China outbreak then. And there were a lot of conflicting evidence or publications about how infectious this virus, the coronavirus, the new coronavirus is based. So some people reported a little above 2, some people reported 4, up to 4, and even 5. And we even saw somebody reported up to 7 or 9. There is a wide range, and apparently, the reason probably for that is just not enough data out there actually for people to really look at it. And so Yifan and we actually took a look at this and say, can we do our modeling and see what's the number may look like. So here is a brief outline for the outbreak early on. So, on December 31, and the cluster of 27 pneumonia cases were reported and detected in China. And then the first death mortality happened about 2 weeks later. And then on January 23rd, that's the city lockdown, and decided to lock down the epicenter 8 hours. The announcement was made about 8 hours before the actual city lockdown. And people are saying that probably 300,000 people actually just fleeing away during that 8 hours from epicenter to everywhere. So, and then on February 11th, there was some update after city lockdown in terms of total confirmed case. Between December 31 through February 11th, there are about 47 confirmed case and about 1,000 deaths globally. And so if you look at numbers then and compared to what we have now, it's basically almost on the par of what's going on in New York City alone. And here is the map, and then in February. And here, and the China CDC actually reported the epidemiological curve. They first provided a report on January 28th. Actually, we waited about a week after city lockdown because we really want to see what's the impact of city lockdown. And at that time, nobody actually knows what the city lockdown really means, because that has not been really used in modern epidemiological studies or prevention intervention strategies to lock down a whole city with over 10 million people. So we waited for about a week, and we saw the epidemiological curve, and then later on they actually updated the studies, and we are able to see more data through February 11th. And
Ying, quick question.
Yes.
So on that graph that you had before, the red and blue, I'm sorry, I read Chinese. What did they mean?
Oh, the red ones were confirmed cases. The blue ones are the ones they call the suspected cases, so which means they are suspected there are probably infected but not confirmed by assay testing. So that's why you see a lot of suspected cases. Yeah.
Thanks, Ying.
And I will discuss a bit later on why the curve you saw probably not necessarily reflect what's really going on in the field. So, and also some people actually posted their so-called augmented onset time from case report times, and to suspect that to model the reporting delay. And we actually draw some illustrative examples here, and see, first you got infections, and then you have symptoms onset. Then you have people went to hospitals to seek hospitalization, and then the hospital will do lab confirmation later on. So here we actually model the whole period in terms of incubation period, and we also model the relative infection distributions, and we put transmission models here, and then put the city lockdown as an intervention through this modeling, and then we assume different distributions in terms of symptomatic infections and pre-symptomatic infections or asymptomatic infections. And then here you are, we have some updated results. And so if without asymptomatic infections, and we estimate the R0 to be at the baseline to be 2.8 and earlier to be 2.7, but with asymptomatic infections, then the R0 at baseline can be as much as a 4. So this is one of the things we suspected is because now to see why conflicting or confusing results early on when people reported R0 through different kind of modelings. And certainly the model assumptions can be different, is an issue. But the other thing is, at that time, we don't have many people really realize asymptomatic transmissions is such a big issue for the new virus. So now we, for anything, if we want to understand to model the new coronavirus, the asymptomatic infections should be definitely to be part of the equation to do that. And also some model estimation here in the curves. And I will just skip those because there's a lot of details in our manuscript. And one of the key things we learned from our analysis is this one. Is testing, and then for contact tracing and the quarantine of exposed individuals. And the testing and the numbers, Suzanne, you just mentioned about that ones. We don't think that the numbers not necessarily reflect what's really going on in the early stage in China in the epicenter, because at that time, everything's restricted by resources, by the number of test kits they have. So probably the numbers we saw actually probably is just another marker of how many number of kits, testing kits available. So there's our issues, and what we would summarize in terms of the activities we have done around this topic is, the bottom line is we are interested in R0. The significance certainly is important to understand the transmission mechanism. The data is what type of data needed to answer our questions, and what kind of methods. We presented a transmission model. And for students, I think it's particularly we need help with is we need help in terms of data collections and verify data, and to do more modeling. And for example, right now, what we really are interested, for example, is to assess what's the effect of social distancing in different states in the US now with the epidemic curves, and how that will be interacting with public opinions or political opinions. So one thing we would like to do is to do natural language processing to extract all the keywords about different states in terms of newspaper reporting, in terms of what they say by the governors, and then we really want to see how the social distancing interacting with all those things will have an eventual impact or ongoing impact on the transmissions by confirmed case or by cumulative cases. So those are the things we definitely need a lot of help, or students' help, or other faculty help to do more modeling. And so our paper we submitted our paper on February 6th, and we didn't get our revision till mid-March. And then we submitted our revision, and it was accepted. Now it's online yesterday. So anybody is interested, and you can look at this manuscript. Thank you.
Okay, thank you for the introduction of this very interesting project.
Something,
sorry.
may I say 2 sentences? Yeah. It was very good. And the test, probably in this country, a lot of us think that we're only ascertaining about 1 in 10 infections in this country, because we haven't tested. So it's not a problem just in China. This is a real problem in this country.
Yeah, to me, there are a lot of insights from what you talked about. It just let me look at the whole situation differently. It's very insightful. Okay, our next speaker will be Deborah Donnell. Sorry again if I pronounce your name wrong. The topic will be Network and Industry Task Force on Clinical Trials for COVID-19.
Hey, so this is Deborah Donnell. I also am an investigator at the Hutch, and let me find the right screen here. I think it's this one, all right? Sure. And I feel like the topic of this, is this right? Hang on, sorry. I think I shared the wrong screen, dang. You're looking at something that's wrong, right? Oh, how do I unshare it?
Is it wrong? It says task force.
Is that right? Oh, I've got the right one. Oh, I got it. And thank you so much. I think that the title of this talk is a little bit the title of subject that was given for my talk is a little bit misleading. So it'll think what I'm actually going to talk about is the work that I'm doing in clinical trials that are in the field and how COVID is impacting them. So it's a different angle completely. It's not about COVID directly. It's about how do we continue the research and look at the impact of COVID on research that's in the field. So this task force is biostatisticians and data management establishing the principles and plannings related to dealing with disruptions in the ongoing trials because of the COVID pandemic. This is a partnership that includes people from the clinical trial networks at NIAID, which are called HPTN, the AIDS Clinical Trials Group, and the Pediatric Group, the network trials, together with their industry partners. And it involves the network data managers and biostatisticians. The focus is trials currently in active recruitment and follow-up. And one of the tasks is there is both EMA and FDA guidance documents coming out, as always true with COVID, I think, on multiple times every month. And they're really dealing with trying to give us guidance for how we plan for capture of these deviations from measures and the decisions that have been made on ongoing trials. So one section of this task force is about what data capture are we doing for people who are missing visits, having protocol violations, missing procedures as a result of the COVID disruption. In what ways should you be accommodating alternative measures and data collection? And much of this is, can we do it in the home? Can we use ePro, or do we need to stop? The goal is risk management of the impact of COVID on both trial participants and clinical trial conduct, including decisions about continuation and stopping of trial procedures. And the second part is like, how are you going to deal with the inevitable changes in your ITT statistical analysis plan to accommodate the disruptions in the efficacy and safety evaluations of the protocol? So that's what the task force is about. And the second thing I'm going to talk about is how, if I can get the slide to move, is, the particular trial where I am doing this work. So we in the PTN are involved in this double-blind trial of injectable cabotegravir versus daily oral PrEP. These are trials that 083 is in MSM and transgender women. We've enrolled 4,500 people. In 084, this is a trial that Jim Hughes is involved in, and it is in sub-Saharan Africa, and we've enrolled over 3,000 women. So we are 50% through the follow-up in 083 and 25%, I've got the numbers wrong, and 25% through the follow-up in 084. So we're right at this, like maximally informative position for the trial. And what's happening is, so what is happening in these trials, we have had to stop enrollment, which is almost complete. The sites vary from being completely closed to conducting restricted visits where we're asking them to target the drug delivery visits, to being disrupted because participants are unable or unwilling to even reach the site, even if it was open due to transportation issues. So there's clearly, for this particular trial, which is delivering an injection and needs to be done at the site, prepared by the pharmacy and done at the site, there's clearly going to be a loss of intervention delivery during the COVID disruption. So there's the data management side. And what I've been really working on is, what are we going to do with this trial? So involved in DSMB and FDA discussions about modifying the primary ITT analysis. I mean, the first thing we really went to was a pretty simple modification to use an Anderson-Gill approach that will exclude this COVID disrupted time. Because, and this is considered to be okay, even by Tom Fleming, because, the reason for the disruption is clearly not caused by the drugs under study. So this is one thing. So it's one kind of like planning, exactly how we're going to do that. I think in our situation, because of how far through the trial we are, and we're coming up on, we have these scheduled DSMB reviews. We're looking at, some slight modifications of the strategy of interim monitoring, and again, having discussions with the DSMB about what is okay to change and what is not okay to change. And then I think the third big topic of discussion is, strategies for restarting after this disruption, or conditions for stopping if the disruption goes on, because we don't know how long this disruption will continue and how bad the loss to follow-up might be, or how differential it might be in terms of the risk characteristics of the people we enrolled, and what the implications of that might be for gaining licensure when the trial is actually over. So another, I will end there, but just another set of statistical challenges that come up as a result of COVID in ongoing trials. I will stop there.
Okay, thank you for the introduction. Our next speaker will be Leah Fisher with the topic quantification of SARS-CoV-2 viral load and diagnostic assays based on UW virology lab data.
Hi. Okay, so I'll be nice and quick. I don't have any slides to share, so I'll just tell you sort of quickly about the idea. Briefly, I work at Fred Hutch. I'm a staff scientist there, and I primarily work on vaccine trials, HIV vaccine trials with like Peter Gilbert and a lot of other people on this call. So, but what I've been working on lately regarding COVID is I've been working with the UW Virology Lab, and we've heard a lot from people like Bets about testing and how important it is. And so the UW Virology Lab has been doing a lot of the COVID testing here in Washington and for the broader sort of community. And you know, the way it works, Bets did a great job of describing it, is that you know, get a nasal swab and you send it off to the lab and they do some science and they run their assays, and then you get returned a binary outcome. You're either positive or negative. But in the interim, what actually happens is that, you know, they're doing PCR to generate and detect, to see if they can detect viral RNA. And that can actually be, you know, it's pretty common to convert that to those measurements, those continuous measurements, into like a viral load. But the way you sort of typically do it is you have sort of known standards, and so you can run them together and you can compare what they are. But since we're in this sort of different setting where the priority is really identifying cases, we haven't been doing sort of the standard things to identify to sort of calculate viral load continuously. So, so really the project is pretty straightforward. We're just taking the existing data that is generated from all of this COVID testing. We're taking the continuous data and additional standards and seeing if we can use all of that and really get good estimates of viral load. So as far as statistically, it's not super complex, but clinically and scientifically, it's really important. We need sort of these continuous measurements to be able to really deeply understand and study associations and talk about disease progression or understand viral load and severity. And there's lots of other reasons, transmission dynamics. So, so I've been working on that with the lab, and I'm sure they'll have lots of other lab-based things, but I think that's all the time I'll take, and I'll let everyone else talk. So that's the quick and dirty version.
All right, thank you so much. Our next speakers will be Rinda Huang, Holly James, and Peter Gilbert. They'll talk about the WHO core protocol for preventative vaccine efficacy trials. Sorry, we can't hear you.
Can everyone see the slides on the screen?
Yeah.
Okay, yes. So this is joint work with my colleagues, Holly, James, and Peter from the HIV Vaccine Trials Network, together with Beth and Tom and other members of the WHO core protocol working group. So this is the project that both Beth and Tom mentioned in the presentations earlier. So I don't think I need to repeat a lot of the background. Basically, the goal of the project is to develop a core protocol as a guidance document for efficacy trials of vaccines to prevent COVID-19 disease. So this is a trial design project that was funded by the WHO. So this is the title of the protocol. So here's an overview of the design of the trial, a prototypic phase 2b/3 trial of preventive vaccines against COVID-19. In the core protocol, we specify the study population would be adult individuals who are testing negative for SARS-CoV-2 but are at risk for COVID-19 disease, because the primary endpoint is COVID-19 disease. The primary objective is to evaluate the efficacy of each vaccine against placebo for the prevention of COVID-19. With vaccine efficacy less than 30%, setting up as in the design null, where the vaccine efficacy is estimated by comparing rates of disease between the vaccine and placebo group. So the trial design, as Tom also alluded to, is an individual randomized placebo-controlled double-blind endpoint-driven trial. We will, in the core protocol, we described sequential monitoring plans for efficacy as well as inefficacy. And then as Tom also mentioned, so the new vaccine regimens could be added into the trial under the umbrella, under the structure of the core protocol. And the concurrent comparison with placebo group or a shared placebo group may be used when we have multiple vaccine regimens on the horizon to be tested in the efficacy trial. So we have lots of students here, so which is great. So we laid out some questions, design questions, analysis questions. So one of the key design questions is what study population to look at, recognizing the challenges of predicting the future course of the pandemic and how we will select sites or settings with high rates of disease with potential course correct study. Of course, incorporating the mathematical modeling, epidemiology studies that other speakers had mentioned earlier. And another design question is the choice of the design, the study endpoint, which Tom also mentioned. In the core protocol, as I mentioned, COVID-19 disease is the primary endpoint, regardless of severity. However, there's certainly a great public health concern in severe disease and/or death. And what about infection? So that's another possibility, because as some vaccines might work against infection, that consequently reduce the disease acquisition. And then another question is the choice of the vaccine efficacy design null. Currently in the protocol, we use 30%, and the study is powered as 70% alternative to at 90% power to look at 70% versus 30%. But there's definitely a balance we need to consider in the design of such trial between possible vaccine efficacy or a minimum vaccine efficacy level that would be that can be done to achieve population level impact that Laura and Beth also mentioned. There are also more work in terms of setting up the right efficacy boundaries, inefficacy boundaries, and monitoring timings, as well as a hot topic about how do we do the randomization, the individual randomized trial. And we know that how do we select, which may be done in a cluster setting. Another possibility would be doing a randomized cluster level randomized trial. So those are just a sample of possible design questions in such a context. There are also some open trial analysis questions for students to ponder upon. First of all, even within the individual randomized trial, how do we account for correlations of endpoints among individuals within a transmission cluster, within a household, or within a community? The second, and then another question is, in secondary endpoint analysis, where we wanted to assess vaccine efficacy in a subgroup of individuals, accounting for the possible incubation period, or accounting for the immune response ramp-up period, which would normally be a couple of weeks after the vaccination. If we just look at individuals at risk after those periods, then we know that the analysis is subject to post-randomization bias, and how do we account for that? That's one question. And also, how do we account for time-varying vaccine effect in the context of possible change in background incidence of disease due to public health interventions at different stages of the epidemic? And also, lastly, human correlates analysis studies, as well as SIV analysis, will be important to answer in the context of such trial. And Peter will take over to talk about SIV analysis. And Hardy is my colleague who worked closely with this on this with me. Do you have anything to add?
Nope, I'll leave time for Peter. Thanks for a great summary, Yanda. Thanks.
May I say one thing? I have some comment on everything. That was great, Yanda. So we find ourselves in the interesting situation over in our little group in BBE, where you're getting the idea there's a lot of trial design powerhouses that NIH has pulled out of the WHO core protocol. And so Elizabeth Brown is involved with NIH in trying to design vaccine trials. And the WHO is a separate thing, but we all get along fine.
Thanks, Beth.
Okay, yes, thank you, Kendrick and others. And so one thing to say, you know, there is really a large amount of randomized vaccine efficacy trial research ongoing at the Fred Hutch, which is done collaboratively with our UW faculty and student colleagues. And so we very much welcome students coming over to the Hutch and talking with us about potential research opportunities. You know, we'd love to have dissertation students and other types of research projects. So suppose a randomized trial is done of the type that Yunda just summarized, and suppose we see 50% vaccine efficacy to prevent, you know, COVID disease. So at one level, we're opening the champagne and we're happy, but we're also saying, well, that won't completely eliminate the pandemic. So we need to get a 90% efficacious vaccine. So one data analysis that is done of randomized vaccine trials to try to improve the vaccine is called sieve analysis. And the idea here is that, so for any vaccine, it's based on some bit of the pathogen, and there's some strains of that pathogen put into a vaccine construct, and the vaccine induces immune responses that recognize those strains in the construct. But the vaccine may not induce immune responses that recognize other strains of the virus that are circulating and exposing people and causing disease. So what sieve analysis does is it analyzes the genetic characteristics of the viruses that infect volunteers in these FC trials and asks the question, how does the level of protective vaccine efficacy depend on the features of those exposing viruses? And over the years, we've had several students work on these projects and develop statistical methods and studied several genetically diverse pathogens. And so on this slide, I'm not going to go through it, but it's showing some results that the group has found where we were able to discover in these randomized VE trials that the vaccine efficacy was better against pathogen strains that were close genetically to the vaccine, and the efficacy was worse against viruses that were far away from the vaccine. And so for coronavirus, the same type of analysis would be important, given that we know that there is quite a bit of genetic variability of the SARS-CoV-2 virus. And basically, if we could discover what parts of the proteome of the CoV-2 virus are important for vaccine efficacy, then what we could do is take the 50% efficacious vaccine and then engineer it to bioinformatically optimally select new strains to put in that vaccine, and then we might be able to get predicted vaccine efficacy of 80–90%, and then that could go forward in iterative trials, and hopefully we'll get a better vaccine. Thank you.
Thank you, Peter and Yunda, for the introduction. Our next speaker will be Lueping Zhao. His topic will be risk assessment of accelerated pulmonary comorbidity following SARS-CoV-2 infection using host immune genetic polymorphism.
Great, thank you. Do you see the screen?
Yeah.
Yeah, okay, great.
Yeah, we can see the screen, but everything is kind of small.
Oh, okay, let me increase this a second. Got it? Yeah. All right, so this is a collaboration with Dan Garrity and Keith Giron and Peter Gilbert on behalf of HVTN. Dan Garrity has a lab basically is responsible for next generation sequencing to genotyping the commonly known sort of immune genes, HLA, KIS, and MIMIK, and so on and so forth. Keith Giron, as you know, that his lab is responsible for coming up with this the fast test for local UW, and his lab has been related to targeting all the COVID-19 patients today. So I guess in the background section, I think a lot of things we can talk about. I just highlight actually 7-8 points. Is that 80% of infected patients just experience minor flu-like symptoms, and 20% are infected they have severe pulmonary comorbidities. And so it is commonly saying that the old patients or immune compromised patients tend to have severe comorbidity. Old patients, that's easy to identify by age. But what I mean by immune compromise, that is what we know. And the point actually dovetail very well to what Tom just said. What actually people really care essentially is 20% of this infected people. Because otherwise, this is a COVID-19, just like flu, right? That's happening, I was talking about it. So the goal here essentially is, can we actually identify those 20% of individuals? So we have the hypothesis, basically is that we we're anticipating that this is with a point exposure to the the virus, the host immune system basically recognize and present and kill infected cell by the COVID-19. And then for subset of individuals, the immune resistance essentially recognized by class 2, for example, will trigger the cytokine storm that basically excessive of the immune reaction. That will lead to basically severe clinical comorbidity, especially pulmonary functions. So that's not the hypothesis. So therefore, we can focus on candidate immune genes, essentially class 1, class 2 MHC genes, as you know, A, B, C, D, R, T, Q, T, P. And then the key is, and then there are a few other RCRGH and FC gamma receptors. Those are the common candidate genes we're looking at. We hypothesize that the configuration of these certain immune genes, the alleles or motifs or haplotypes or any other configuration probably are predictive of this 20% individuals. And if now hypothesis is being demonstrated sure, then we probably should be able to identify immune prediction models. And so that's the goal of that hypothesis. Now, as of March 30, at UW Medical Center, we have a little over 3,200 of the patients who tested COVID-19 positive. Now, actually, this study that we actually designed really is assuming you will get infected, really the amount of infected individuals, we want to know whether the immune system actually different between those that have severe symptoms versus not. In this table, basically across all the columns, you have the inpatient, basically the request of COVID test is come from inpatient, and of course from an outpatient care, and emergency employee and unknown. And at this point, because of the time limit and so on and so forth, those are all in the electronic records and scattering around the Seattle area, some of those Mayo Clinic. It is accessible, but we don't have time for this at the moment. We're basically assuming at the moment, the patient, inpatient, those tend to be the patients that require hospitalization. Whereas outpatient, those are probably drive-by and so on. So those probably tend to be just infected individual, have flu-like symptoms. So if you look at the genders, so between the male, female compositions, and among the inpatient, then you have more males, and the outpatients, and a little more females. And across the age groups, from the ages 0 to 30, 30 to 40, and so on and so forth, you can see that people across all ages get infected. So this is basically is an illustration that people do get infected when you're exposed to the virus, and it doesn't matter what, regardless of the age. Reality. Now, within this context, we basically can focus on those inpatient and treat those as if they probably tend to have more severe clinical comorbidity, and outpatient it tends to be less. And the question basically for our pilot study essentially is that can we try to answer the question that can we actually identify genetic profiles of immune genes that actually really separated those two group of patients. And that's the pilot study. We're hoping to generate a sufficient amount of the preliminary data to show this one. This is a very different type of genetic test as opposed to the serology test or antibody test and so on. So this is more genetic test. Essentially, it's to really identify the individual at genetic level who are at a high risk for severe clinical symptoms. Okay, so this is kind of getting back to what Tom was talking about. What people really care, essentially, that after getting the COVID-19, whether you can be really have severe clinical symptoms and therefore you require hospitalizations or require the extensive treatment and possibly fatality. So then what we expect of, essentially, then we are anticipated among the several of the key genes we're looking at, especially the HLA genes and also the key, we kind of really have very high hope to identify some of the genes. And then, of course, once you find a gene, then we will be able to find those actually gene alleles and therefore corresponding proteins. And then once we can identify those proteins, then obviously then we can integrate them to create a single response. So there are many different ways to do that. So that I probably don't need to elaborate. So this is in terms of data generation was actually everybody's really hard, and for both from the lab side as well from the database side for HBTM. So all the data now come together. We actually now just have some of the biospecimen have been sent from the Jerome's lab to the Kennedy lab. So then we actually see several different potential applications if we actually could have this kind of response. One is that to really be able to identify some of the low-risk individuals, at least susceptible to severe COVID-19 infections. With or without detector, right now we're talking about a fast test to detect people who have antibody. If with genetic test, even people don't have antibody, you will know someone can be at a low risk or not. So I think it has implications for really identifying people who can actually continue to do the essential work, like military, like in some of the essential department. So I think that would be very important, identify those individuals. And then for some time, the flip side of it, it's equally important, besides age, we also want to identify high-risk individuals. Then if you know someone really actually at a high risk, the moment they tested the COVID-19 positive, you really probably need to get the early clinical management, because those are the individuals at their high risk, besides age. So I think we actually have some incidents that have heard about it. Someone aged 30, suddenly they died for no obvious reason. So those are the individuals you really want to catch them early. And of course, then the next one is that I think that right now it's too premature, but if we actually have this kind of test, and I think it may be useful to think about it, develop vaccines and guided by those responsible immune genes. Because with this particular virus, we can actually really identify which specific antigen peptides and really bombard and recognize. Maybe that we can be developing the vaccine very specific target. This is 20% individuals. And of course, then this is also by same token that you really want to look for this compounds for immunomodulatory therapy, specific for prevention and intervention for this high-risk individuals, as opposed to for entire population. I think that regardless of what sort of compound you use, if you for entire population, there may be some unintended consequence. But if you can identify as a high-risk individual, so I think that maybe it will be more effective. And for that reason, I think then if any students will be interested in thinking about this, we'll be looking forward to some of the help. Think about it, especially we're actually going to move forward to do some more preliminary data analysis and then future proposals. Thank you. That's it.
Thank you, Lui-Ping. Our next speaker will be Paul Olafson, and he will briefly talk about immune correlates analysis, drug evaluation, and basic science of SARS-CoV-2.
Okay, thank you. I have no slides to share, so I'm not sure if I'm being seen, but hopefully I am. So let me just cut to the chase here. What I'm doing is I'm collaborating with long-term collaborators in the Seattle area and outside of the Seattle area. In particular, right now, what I'll talk about is work with Mike Gale, who is doing a lot of transcriptomics for immune biology at UW, and Louis Picker, who's at the Oregon Health Sciences University doing a lot of immunobiology and vaccine development. And what we're doing is we're accessing samples from Seattle area, and we think also Portland area people who have had coronavirus 19 and who have the different outcome groups, mild, severe, death. And we're going to be doing a more immune correlates assessment. And what we mean by that is we'll be applying some of the same technologies that we've been applying for our vaccine work and other work. So particularly ICS, which is a T-cell assay using flow cytometry, serology, which will be really important here. We'll be looking at transcriptomics, in particular ATAC-seq and RNA-seq, to look at trained immunity. There's a lot of evidence that innate immunity will be relevant here. We're also going to be including clinical covariates, clinical features. And our goal is to understand the basic mechanism here and what differentiates people who are protected from not protected, following various leads that are really pretty exciting to follow and talk about. But this is, of course, very urgent. So the bottom line is that we're doing immune correlates of disease outcomes and treatment responses. We're working with drug companies that are supplying drugs that we're testing in the lab on in vitro cell lines, as well as on cells from people. As was mentioned before, and I don't think I need to say it again, that was a great presentation we just heard with a lot of the same things mentioned. So this will be important for vaccine development, treatment development, knowing if folks are no longer susceptible. I just want to mention that antibody-dependent enhancement is known to be the case, and this is a very important issue. Just having positive serology should not mean that we should assume that we're protected. This is an important issue to understand. Antibodies against parts of the virus that seem to be enhancing. So what we're looking for, if we're looking for help, which, you know, help is good, from a statistician, from a biostatistician helper, would be help on these areas of basic assay statistics and designing these studies in vitro and using the samples wisely. It's pretty basic statistics, but there's a lot of areas to be developing new statistical methodology. It's mostly focused on getting it done. If students wanted to help, if you want to help, I think that would be great. What we would need from a student to help would be someone who has a lot of passion and self-direction and patience and is really good. So if you are that person, please contact me. Thanks.
Thank you, Paul. So last item will be the topic survival analysis with AA interval censored and truncated data for individual COVID-19 infection data. The speaker will be Gary Chen, Fei-Gao, and I will also briefly introduce my work.
Okay, so hi everyone. So for those of you who do not know me, I'm Professor of Biostatistics at UW, and I am an affiliated faculty at BIT. But I also have a joint appointment at Health Services at UW. So, well, I guess I'll first divert a little bit before I talk about survival analysis, because I'm actually also involved in some policy analysis, because I have some connections with health policy researchers and actually some state legislator who sometimes want to, evaluate, whether what they sort of place as policy, whether that is sort of, helpful or not. And also back in January, when the outbreak is still sort of in China, Andrew Zhou, which was one of the UW faculty, he's now chair professor at Peking University. He was in Seattle at that time, and then he actually asked me and one of my students, Fansha, to join his team working on some policy evaluation for the Wuhan lockdown. And I think maybe Fan can sort of share about this in a future presentation, but for now, we would like to talk a little bit about the individual infection level data that people have been collecting, which has some pretty interesting survival analysis type of problem, because the infection data, actually we can't really identify the sort of dates of infection. We probably can only sort of identify, say, a range that a person might be infected. For example, using traveler data from Wuhan that's sort of traveled outside Wuhan, and then we can sort of look at when they are so in Wuhan and during the early outbreak, and then to sort of limit the the infection interval, but it's never sort of identified, right? And then so this is a problem that you have interval censoring at the left endpoint, but you also often have interval censoring at the right endpoint, because the say symptom onsets, people also might sort of report that they might have onset like at a certain time period. So so there might be some interval sensor at the right. So this is a kind of doubly interval sensor data. And also there's a truncation issue with the data, because for travel data, they have to travel outside Wuhan. So actually the time from infection to travel is a truncation time. And then in general, you are actually getting people with longer incubation period, because there's a selection bias that's going on. So that's sort of like why this type of data is doubly interval censored and truncated. Well, this is sort of like an overview, but then Faye and Kendrick will work on different aspects of this data, and then they are going to share about software. We will start with Faye.
Oh yeah, thank you, Gary. So my involvement was actually not that complicated. My work is to develop a better model to estimate the incubation period. So basically, you don't know the exact time between you're affected and you have the symptom onset. That is the time that we want to estimate. But as Gary just said, we have the interval. We only know the lower bound and the upper bound of the infection date, and maybe the lower bound and the upper bound of the symptom onset date. So we need some parametric models to estimate the incubation period, which makes more sense. I think, so in the past, I read some current literature. Most of the results, they borrow some values from the past literature, which assumes the incubation period is known, like 14 days, but it's like chicken egg. You use the results from some literature to derive the results of other literature, and they're kind of interdependent. So it doesn't really help to answer the question cohesively. So that's what I'm doing here. I want to joint model the incubation period and the infection time so that we can get more reasonable results. That's a very brief overview of what I'm doing.
So maybe Faye can also talk.
Yeah, sure. Yeah, so first to introduce myself, so I'm Faye Gao, and I'm now assistant member at Hutch. So I'm working on the non-parametric or semi-parametric aspect of the problem of doubly interval sensor data. So previously we have been working with the single interval sensor data where there's only one endpoint that's made subject to interval censoring, and we have used the Poisson augmentation idea, which is trying to link the process of the event time as a Poisson process so as to ease the computation to propose a very normal EM algorithm. So now we're looking at whether we can apply this type of idea to this more complicated double interval sensor data. You see, like it's where both the starting point and end point can subject to interval censoring. So it's much more complicated. And since right now we cannot really determine where the survival distribution will take jump, and we're trying to see if we can apply similar idea to this setting where we can look at the probably non-parametric estimator of the survival distribution or semi-parametric approach, trying to see the effect of some covariates such as gender, race to the distribution of the incubation time.
Okay, thank you. Just want to make sure, is that all? Is that the project? Is there anything else? Okay, all right. Then I think, yeah.
Yeah, so maybe now we can open up the floor just to a few questions, and then from there we'll probably take it offline and maybe just email the investigators. But maybe we'll take 3 questions before we close out.
Yeah. All right. So I just want to mention that other 2 students also help with keeping the minutes and keeping the questions. I just want to mention Subodh and Adam. They're graciously willing to help. Okay, let's go into the question session.
I have a question, if that's all right. Yeah, so I think this relates maybe more to some of the monitoring and modeling of the forecasting of the outbreaks. In the news, there's been a lot of discussion of plots of reported cases, usually on a log scale versus time, and comparison between the US and say Italy, et cetera. But of course we have very different testing regimes in different countries. And I'm wondering if anyone has a comment on that and how we can meaningfully interpret these numbers that are probably generated by very differently censored data mechanisms.
Okay.
I think I can comment. Well, I'm not very sure about like difference in terms of like testing capacity and other things, but like there are, say, in policy analysis, people actually are looking at, say, difference in the policy, because the policy, I mean, they are also, well, a lot are social distancing, but they actually have like heterogeneity, like, you know, where those, policies are sort of implemented. So there are some policy researchers who are working on sort of defining the exposure, basically the policy exposure by like scoring them. But this basically is more like the policy researchers that are doing.
This is Suzanne. I have a question for Deborah, maybe for others as well. I was particularly concerned with the interruption on existing trials, that there could be a specific bias towards not getting sufficient safety data, because if one of the treatments has a safety issue, but we don't collect enough safety data for a period of time, even if the main results are somewhat biased as well, I'm much more concerned about biases in safety signals, because we have a break and that might be tilted towards one treatment and help us or have big concern in not detecting a safety signal that otherwise we maybe would have.
You are right to point that out. It could be extremely difficult. I mean, if you could imagine, for example, if your outcome was death and then you have a COVID-19 pandemic, how could you sort out what the cause of the death was? It would be very difficult. I mean, I'd say the only other consideration about this to keep in mind is that it is clear that it is you know, sort of an external event that is causing this, that it would be difficult to you know, there's nothing about a person's participation in the trial that would lead to the data being omitted, because my intention, for instance, in my trial is to omit events that happen while COVID-19 is disrupting the trial. And I think if death was your endpoint, you might have to do that as well. And because there certainly is a possibility that data would not be reflective of the safety of the product. So, you know, there are some very difficult issues. And I think every trial, depending on its population and its endpoint and the potential for COVID-19 to differentially affect the outcomes that might be observed in that period or even after that period is one of the very tricky things that we are going to have to confront with these clinical trials that are in process. I know, Tom, I'm sure you can comment on this because you are being consulted as a DSMB chair in some of these issues as well. I don't know whether he's still on.
Yes, yeah, and this is a difficult issue, particularly if we have an immune suppressive intervention that could in fact have positive effects through that mechanism in the normal disease process setting, but could have amplified unintended negative effects in the presence of COVID-19. So we are left in studies that are ongoing with the challenges of trying to understand in protecting the integrity of those studies. First and foremost, how do we safeguard the interests of study participants if the procedures that are experimental in the study lead that person to be more exposed in violations of social distancing? How do we maintain adherence that would be standard real world adherence in the absence of the pandemic? And how do we attain or how do we maintain retention? And yet, even if we do all of those based on what both of you are saying, and you're right, if COVID-19 interferes with the actual true benefit risk profile of that intervention, then we're going to get results that are not relevant to a real world setting outside the pandemic.
Yeah, thank you very much to both of you. I think multiple imputation and missing value considerations will play a much more important role, not only with regard to impacting the mechanism, but just the ascertainment of these events. That because if we don't get the in-person follow-up there, and that might be more biased in some sense. So I think the prospect or the issues surrounding missing values is going to be even more extreme here, probably. But thanks, yeah.
So I have a question. There's a white paper out there, I forget by whom, arguing for the ethics in a vaccine trial of deliberately exposing trial participants, with informed consent and all that, to the virus. So I guess I wonder what the experts here think about that, and I am also curious whether that idea is has gained any traction.
It's not part of the thinking, at least in the mainstream development by WHO for their core vaccine trial.
I guess I would ask, is in the midst of this pandemic, why would you need to? I mean, it certainly seems ethically difficult. And I don't think you're going to have a lack of cases in any trial that you do.
I agree, Deborah. I think that the ethics of it are, and Tom, I think the ethics of it are completely wrong given the amount of exposure that people are going to see in the near future in populations.
I actually had a question for Laura about the modeling work that you did about delaying the epidemic or flattening the curve. Mm-hmm. And my question for you was, the metric you were looking with, I think, was something like number of people who got infected, but yes. But my thinking, the flattening the curve, my understanding was that the goal of flattening the curve was to not overwhelm the hospital systems for people who were, for people who needed medical care. So it's so like particularly if you look at the impact of 60-year-olds and above getting getting COVID infections, that would have a much higher impact on hospital impact. So it just seemed to me more relevant to model the impact of these cases on the healthcare system rather than actually the number of people who get infected.
Right, and I have a figure for that too, which of course I closed, but I did look at that too, and so here you can see my screen, correct?
Yes.
Yeah, so I did a pretty bad job of explaining things, but so really briefly, in this I was comparing the left thing is what I think happened in Seattle, given the timing. And the right thing is what might have happened, say, in New York, where they started their interventions where they were already in the exponential phase. And so you see that the effect of flattening the curve doesn't really happen in the left panel. You're only delaying the epidemic, and it will as soon as you open up the interventions, that you will have the epidemic will come back if you don't do anything differently. And so coming back to your question, well, in the other case, you do see a flattening the curve. So there was all this buzz about that word, and I wanted to point that it's not necessarily true that you're gonna flatten the curve regardless of where you start. It really matters when you start doing things. And regarding your point, you can see, so this plot is slightly similar, but here I focused only on the 50 days. So that this will be more like the Seattle scenario where you started your intervention right at the beginning of the epidemic. And so this green thing refers to what you're saying. So this is proportion of cases averted, and this is proportion of hospitalizations, and this is proportion of deaths. And you can see my mouse, I hope.
Yes.
And so what you can see is that, yeah, when you only protect the high-risk groups, so people age 60 and older, you can achieve a really good, you could avert almost 40% of the hospitalizations and over 40% of the deaths. Of course, this is a very idealized scenario, so you have to, I would not like you to take these numbers with, you have to be cautious when you talk about these numbers, but you could achieve a lot by only protecting the high-risk groups. So yeah, I did look into that. But I should say this is considering that I'm only looking at the first 100 days. So obviously, if after the first 100 days, so after the 6 weeks that Governor Inslee gave, you come back and you open everything up as if everything was before, these gains, you will lose them because those people that were not infected then will be infected afterwards. Does that make sense?
Yeah, it does. Thank you.
Yeah, you're welcome. Thanks for asking.
All right. Thank you for the questions. I guess that's all the time we have. So if you have if any of the audience have more questions, contact the speakers by email, or you can also send me an email with a question. I can forward your question to the speakers. So I guess that's all the meeting. I really learned a lot from the meeting, so thank you.
Thank you.
And we went a little for the audience for keeping with. Is there, yeah.
There's one last thing we will say is because today we are really much compacted and a lot of things we cannot talk in details. So we are thinking about more sessions with more details upcoming. So please stay tuned and not only for our students currently enrolled, but also we're going to have topics and for our prospective students to see, Seattle is not epicenter anymore. So, and but we have so much exciting research going on. So that's the other thing we are contemplating, we're planning. So stay tuned.
Okay, thank you. So I guess that's all for the meeting.
Have a nice weekend.
Well, thanks so much, Kendrick, Alex, and Michael, and Peter, Tom, and everybody.
Yeah, thank you for coordinating this. It was great.
Yes.
Thank you.
Bye.
Yeah.
Bye-bye.
Bye, everyone.
Bye.
Have a good
Bye,
weekend.
everyone.
Stay safe.